disorders, optimizing differentiation protocols to produce functional muscle cells and brown fat organelles.
Thank you for being here and attending my presentation. So I'm going to present my PhD work, which is on the synthesis of iPSC, so induced pluripotent stem cells derived brown adipose tissue in vitro. So brown adipose tissue is known in human and also in other species for being involved in non-shivering thermogenesis. And in human, it was mainly known for its role in thermoregulation in newborns. So we have like big brown fat deposits between the interscapular region, in the perirenal region, perikidney region, and in the periaorta region. So until recently, we thought that the brown fat was disappearing during adult stages, but 10 years ago, we discovered that this tissue is remaining in the adults in small deposits. So the interest of this tissue is mainly due to the UCP1 protein, uncoupled protein 1 protein, which is in the inner membrane of the mitochondria. So in the inner membrane of the mitochondria, we have what we call a respiratory chain, which is basically a complex of different proteins that create a proton gradient, which allows to generate energy. So in most of the cases, we are most interested in the ATP generation through this process. But the uncoupled protein 1 will uncouple this respiratory chain and use the proton gradient to release the heat instead of creating ATP for storage of energy. So the brown fat tissue and brown adipocytes, which compose this tissue, are very rich in terms of mitochondria. So they consume a lot of glucose and fatty acids to generate the heat. So which makes a very strong interest toward this tissue because it is a potential therapeutic target for metabolic disease, obesity, type 1 and type 2 diabetes. So my specialty is more on the development side than the physiological side. So from a development aspect, the brown fat comes from the mesoderm. The mesoderm is one of the 3 layers that compose the embryo during the gastrous stage. So 3 layers are the ectoderm, which will give rise to neural system and skin. The endoderm will give rise to gastrointestinal system, and the mesoderm will give rise to several tissue, including brown fat. So this mesoderm layer will be segmented during the development and will have different sections. The first section will be the paraxial mesoderm, which is the closest to the neural tube. Then we have here the intermediate mesoderm and the lateral plate mesoderm. So our interest will be on the paraaxial mesoderm, which is here. So this paraaxial mesoderm will segment itself during differentiation in the antero-posterior axis and will give rise to another structure called somites. Somites are like epithelial balls that will be segmented themselves into 2 sides. The sclerotum, which will give rise to tendon, cartilage, and bones, and the dermomyotum, which will give rise to dermis, muscle, and brown fat. So from the single cell point of view, if we start from pluripotent stem cell, we'll have different brown fat lineages. So on the left, in green, we have what we call the MIF5 negative progenitor, which comes from the lateral plate mesoderm, the one that was in the extremity in the previous slide, which will give rise to 3 types of adipocytes. The yellow white adipocytes, which is the basic fat cells that we know for storage of triglycerides and energy in general. We have the pink adipocyte, which is a form of adipocyte used for lactation during pregnancy. And we have the beige adipocyte. The beige adipocyte is kind of an hybrid between the yellow and the brown adipocytes. He can switch from the phenotype of the white adipocytes just for storage. And under stimulation, it can express more UCP1 and have a brown fat phenotype. So on the right, we have the paraxial mesoderm lineage, which is characterized by MIF5 positive precursor and also PAX7 positive precursor. And this PAX7 positive precursor will give rise to 2 cell types. One is the brown adipocytes, which is characterized by a pre-adipocytes GATA6 positive and UCP1 mature brown adipocyte. On the right, we have the classical like skeletal muscle cells. So the goal of my team was to reconstruct in vitro the brown adipose tissue from induced pluripotent stem cells for future cell therapy perspectives. So maybe you don't know what is induced pluripotent stem cells. Induced pluripotent stem cells are cells that were reprogrammed. So basically we take mature cells like skin cells, muscle cells, and then we treat them with what we call the Yamanaka factors, and we completely reprogram them to have a complete naive cell. And cell therapy basically is to take this iPSC and to differentiate them into a tissue that we want and inject them in humans. It allows several things, including the unlimited amount of cells that we can generate in vitro, potential autologous engraftment, and it overcomes different difficulties, including the pharmaceutical treatments, because it can lead to toxicity and low specificity during treatments. And also it allows us to benefit from all the metabolic effects from the brown fat and not specifically one protein that we would enhance also. So my lab successfully generated brown fat in vitro in 2D plates. It was published like last year, and the single cell RNA sequencing data showed that we had different population. So we have 3 population in the center here. This is a UMAP representation. So in this representation, if 2 points are close to each other, it means that their transcriptomes, meaning that all the genes that are expressed are close to each other. So the cell identity is close to each other. So we have the cluster in blue, which is a cluster of progenitor that will differentiate into the brown cluster, which is basically brown adipocyte precursors, and the orange one is mature brown and functional brown adipocytes. But this method allows also to generate cartilage, smooth muscle, and neurons. So outside of the fact that the cultures are not clearly pure, the main limitation of this for cell therapy purposes is that according to Liu et al., which is a paper published in 2019, brown adipocytes are extremely weak structures. So when we dissociate them to engraft them, we have more than 97% death in the first week, let alone the 3% that will barely survive. And in my lab, we tried these experiments and we concluded the same thing, that the brown adipocytes are very weak. And when we dissociate them and inject them into mice, the cell die. And after like a month, we cannot find any human cell. So my project was to generate brown fat organoids that are in 3 dimensions and that are floating cultures that allows us to directly graft this tissue without having to dissociate it. And the purpose of this is to transplant them into STZ-induced diabetic models mice. So basically, we inject STZ in mice to destroy the beta islets, so the mice will not secrete any insulin. So we are in like a type 2 diabetes, and then we engraft potentially the organoids that we generate. So the first part of the project was to generate those organoids. And in my lab, we have a model that we work on, which is called somatoids. So basically, these are organoids of somites. Of course, yeah. So these are organoids of somites. We have the protocol already published, and this allows us to generate organoids composed of several somites all aggregating to each other. And we take this, since we know that brown fat comes from a somatic lineage, we just develop the rest of the protocol to go from the somite to the mature brown fat. And these are very, very long protocols, which take 60 to 90 days to have a good amount of UCP1 positive cells. So here I performed the immunostainings of these organoids that we generated with immunostaining of anti-mCherry, because the line that we are using to generate those organoids is UCP1 mCherry. So the mCherry is linked to the expression of the UCP1. So what we can see here is that at day 20 of differentiation, we have almost no UCP1. The first UCP1 cells are generated starting from day 40 of differentiation. And at day 55, we have accepted amount of UCP1 positive cells in the periphery of the organoids. And only at day 75, we have a large amount of UCP1 positive cells. So the signal that you're seeing here is nuclear because the sequence that we are using is UCP1 mCherry NLS. So which means that when UCP1 is expressed in the cells, we have also T2A sequence, which will allow to cut the M cherry, and the M cherry will be redirected to the nuclei. So what we observe here are positive nuclei. This is just a higher magnification. So also this is a body pie staining to show the lipid droplets formation, and which follows the same pattern as the UCP1 cells signal. So the first positive signal, so the first lipid droplets are observed at day 40, and those droplets are very small. And then the number increase at day 55 and 75. So to have a clear quantification of this M-cherry UCP1 signal, I performed nuclear extraction, and those nuclei were then purified and sorted to have the percentage of M-cherry positive nuclei on the total W positive nuclei. And what we can observe, this allows us to track like the expression of UCP1 across time and differentiation. So what we observe is that we have a peak of expression of UCP1 in our model of 30% at around day 60. And the more we do long-term culture, the more the percentage of UCP1 is decreasing. That we don't know why. So to further characterize this model, we performed single nuclei RNA sequencing. So we performed single nuclei and not single cell because, as I mentioned, the adipocytes are very weak structures. So we just destroy everything, we capture the nuclei, we purify them, we sort them, and we sequence them. So this is day 20 of culture. So this is very early stages of our cultures of organoids. And what we can observe is that we have a very big cluster here, the cluster number 0, which is composed of brown adipocyte early precursors that most likely express like fibroblast genes. We have also maybe like almost half of the organoids, which is composed of cells from the muscle lineage. And we have like some small clusters, the cluster number 4, which is dying cells, and the cluster number 3, which is neurons. At day 40, the cells are more engaged toward the brown adipocytes lineage. What we can see is that we have two massive cluster of precursors, the cluster number 1 and the cluster number 2, that also display fibroblast features. And those two clusters will differentiate into the cluster in blue, the cluster number 0, which express already very clear markers of brown fat differentiation, as for example, PPAR gamma C1a. And this cluster number 0 will then give rise to this small cluster here, the cluster number 5, which express AdipoQ and UCP1. So this is our like mature brown adipocytes. And again, we have other like peripheral clusters, but very small, of proliferative cells, of muscle lineage, and of neurons. So we go a bit further, day 55. At day 55, we can see that the cells are more and more mature. So we have here in blue, in the green, the cluster number 2, which is composed of PAX7 peridium 16 progenitors, which are also very, very early progenitors, which means that these cells can still give rise to either muscle lineage or brown fat lineage. Then those cells will differentiate into the cluster number 0, brown adipocyte early progenitors, mainly characterized by the marker PDGF-alpha for fibroblast, but also EBF2, GATA6, which are specific to the brown fat lineage. Then we have the cluster number 1, which is mainly composed of brown adipocyte expressing PPAR gamma C1a, UCP1, and other brown adipocyte markers. Interestingly, at this stage, we observed the cluster number 4, which is mature brown adipocytes, but stressed. Because the more we wait during the culture, the more the adipocytes become big, and the more the cells that are in the center get stressed. So they express stress markers like hypoxia and other markers. Finally, the latest stage that we sequence is the day 75. We can see that most of the cells are really engaged at very advanced stage of differentiation, with the cluster number 4 and 0 as early precursor cluster, and the cluster number 3 in red, 2 in green, and 5 in the brown that are very, very mature. And again, we can see that at day 75, the organoids became bigger and there is much more stressed cells. So I used ScanPy on Python to treat this dataset, and I concatenated all of this dataset that I showed you. So we have in this figure all the cells from day 20 to day 75 of differentiation. So we can clearly see the differentiation process. So this cluster here, the cluster number 6 is the earliest cluster. So it expresses BAC7 and can give rise to either brown adipocytes or muscle. So if you follow the path on the right with the cluster number 5 in brown, you will get muscle cells. And if you follow the path on the left, you will go through the different steps of brown fat differentiation. So then the in vivo experiments. So we engrafted these organoids into STZ injected nude mice. So these mice, first of all, are immunodepressed. And so because we cannot graft human cells into mice that are immunocompetent, they will just completely kill our cells. So we used immunodeficient mice, and then we destroy their beta islets to stop the production of insulin, and grafted our organoids to rescue their phenotype. So what we can see here on the left, so on the left, this is like the 12 first week, and on the right, the 12 first months. So we follow these mice on 1 year. We can see that we have a 60% rate in rescuing the glycemia. So here on the y-axis, we have the blood glucose in milligram per deciliter, and the axis, this is the time. So we have 9 successful transplants that completely saved the phenotype of diabetes. And we can see that the glycemia is reaching levels that are very close to the normal controls. 6 of these mice transplanted did not succeed, so we don't know yet why. Maybe the transplant didn't work, it was not integrated to the host organism. And we have glycemia levels that are still very high. However, that's why we followed these mice on the very long term. We can see that even the failed mice, if we follow them after more than 3 to 4 months, even those mice will see their glycemia level decreasing. So we also performed glucose tolerance tests. So these mice, during like a short period of time, we inject them glucose, and we see how their organism is able to regulate this high new amount of glucose in their blood. And we can see that the mice that were successfully transplanted had very close results compared to the normal control. So they were able to metabolize this glucose and prevent the blood glucose levels to be too high. Finally, this is just a bit of histochemistry on the engraftment that we performed. So the wall here that we can see on the left picture is the organoids that we engrafted. And as you can see, the center of these organoids had to, we can observe clear necrosis, so this tissue was dead, but the periphery of the tissue was able to integrate the host organism. And UCP1 staining show that we still observe a lot of UCP1 active cells after the transplantation. And this tissue was collected 6 months after the transplant. So 6 months after the transplant, we still observe functional brown adipocytes. Thank you. Do you have any question? Yes.
Okay, we are running behind the schedule. There's a space on for 1 or 2 questions.
You said on one of your earlier slides that 9 out of 15 mice, they recovered from diabetes after the transplant. Was that type 1 diabetes or type 2?
Excuse me, have a problem with the computer. What did you say? Is this the type 1 or type 2? So I guess it's type 2 because we prevented the, no, this is type 1 because we prevented the production of insulin. In the type 2, I think the insulin is still produced, but we had lost completely sensibility of the impact, the peripheral tissue will not be able to process the insulin. Any other question? Yes.
Hopefully that will be the last question. Sorry.
It was wonderful and it can be very meaningful, kind of that we can take it from thanks to the bedside. What do you expect? I'm sure there are going to be many things that needs to be kind of sorted, but what are the main things that you think will require to go to the bedside?
Excuse me, what can you repeat?
The human you know, to take it all the way to human.
I see, yes. So first of all, we need to go to clinical tests. So I think we first need to improve our characterization of what happens when we engraft our organoids in mice, because we need to work on whether the organoids will be innervated, vascularized, and why do we observe this decrease in blood glucose? Do we observe also metabolic effects from the secretum of our organoids? Before going to humans, we need to characterize very well what's going on in the mice. Once this part is done, then I guess we would be ready to go to clinical trials on humans.
Thank you. The next session will be